Hi, my name is Julian, and today I'm going to be talking about the ethical implications of human brain organoids in clinical research. Why do we study brains? Because they're everything we know. They make up our entire understanding and perception of the world around us by taking in information, processing it, and storing it in different ways. And based on the different ways that it's stored, it's perceived, and we act upon it differently every single time. Our brains are what determine how we respond to the world around us, and they're made up of different regions that serve various functions in establishing mechanisms of cognition and behavior. Those specific regions can be isolated in humans and in animals to determine their specific functions and how they operate. The various regions of the brain that we can isolate are called organoids. They refer to a variety of self organizing cellular structures that evolve into brain tissue, which represent the developmental and functioning of both atypical, typical, and disease states within the brain. These states can represent the whole or specific regions of the brain as well. Because they're a region of the brain isolated from a human body, it's hard for them to survive without vasculature, neuronal signals, and a microenvironment. So they're integrated with animals and placed into their brains so they can slowly adopt their vasculature and almost take over that area of the animal's brain over time. They've proven really effective as a 3D model for research because they can be generated using fully or partially organic materials and observed through experimentation. Using them as a model for whole brain functioning has an advantage over traditional 2D computational and planar, planar cell models, animal models, and isolated non living tissues from humans because they can be kept alive and observed as they grow and mature. Depending on the materials used to generate the organoids, they can model specific mechanisms of disease and specific functionings of neurological disorders in different regions of the brain. Because they integrate with animal systems so well, it's thought that they're a better alternative to using extensive genetic manipulation in animals. As we know, brain organoids can be used to replicate human functioning and physiology in a model that is replicable like an animal system. The primary argument for using organoids in research is the potential to create a system of individualized care. This is where organic material could be taken from a patient, transplanted into an animal model, and studied separately without having to undergo clinical trials or testing medications in that person. This is incredibly important because it could revolutionize the way that we treat detrimental neurodegenerative disorders. These models have already shown the potential for successful treatment of cystic fibrosis. There was a positive response to the novel and costly drug ivacaftor that was observed in gut organoid cultures from an individual. The positive response was enough for regulatory agencies to approve this treatment as a viable therapy for the disease. Cases like this have given cause for the pursuit of lower risk testing using organoids in place of human trials or in addition to animal models. This could eventually lead to the development of effective individualized treatment for neurodegenerative and neuropsychiatric disorders and create a new standardized system of care if a concrete method of experimentation were to be put in place. Broadening our research could give us a much deeper understanding of such devastating conditions like Alzheimer's and Huntington's disease, and could make strides of improvement in not only clinical research and individualized care, but also in the healthcare system as a whole. While there's a lot of ifs in this method and what it has to offer, it's important that we acknowledge that it has the potential to grow and expand as technology does, offering us much more in the future than we can predict. While there's a lot of evidence in support of organoids in research, there's a lot missing from the ethical side of the argument to support them. Before I get into anything about the models themselves, it's important to talk about informed consent, a topic we've discussed in class and is incredibly prevalent in medical research today. The biomaterials used to cultivate these models are taken from either patient donors who consent to donating material, or they're taken from biobanks where tissue is anonymously donated and used for future research. Because this tissue is used for stem cell derivation to generate organoids, there should be an explicit agreement between patient and researcher. A donor must permit their biomaterials to be used in this form of research specifically and no other. Especially in the case of patients who are too young to decide for themselves or a disabled adult, a trusted legal guardian should be fully involved in the informed consent process. All parties, including third party donors and researchers, should be involved so that biomaterials can be used to create brain organoids and their understanding can be assessed throughout the entire process. On top of that, it's also possible for organoids to develop some semblances of consciousness. It was shown in an in vivo model that an organoid connected to a computer system was able to interact with a simulated world and play the game Pong. In doing this, it was able to prove that it could interact with its environment and make judgments based on the stimuli it was presented with. While there's much ethical discourse over whether or not it should be considered sentient, it has not met the qualification for consciousness in being able to identify or distinguish itself from the environment around it and form ideas and beliefs of its own. So it hasn't yet been considered a. Because this system has the potential to cure diseases and pathologies in people, animals, and anyone else, it's also possible to do the reverse, to enhance them beyond normal functioning in ways that could give them an advantage over other people. Because this technology is so emergent in the field, it's unpredictable. There's no way to know or gauge the effect that it will have on someone genetically by implanting an organoid or systematically affecting their behavior and how they interact with the world. It could change someone's personality. It could change the way they act. I think organoids in research have the potential to deepen our understanding of neuroscience and brain physiology and revolutionize the healthcare system. It has an incredible amount of potential for both good and bad. I think it should remain regulated as technology advances, observing the ethical implications of it and the technological implications of it as we advance in healthcare and medicine. I think its potential to change people's lives for the better outweighs its moral drawbacks because regulations to keep it in place and safe for other people can always change as it develops over time. Thank you for watching, and I hope you learned something.